first vegetation map on the Antarctic Peninsula. Actually, this is over-exaggerates the amount of vegetation there, because if I actually show this map with just the pixels that were vegetated, you wouldn't be able to see them. They're too small. So you have to give some slight exaggeration, but it gives you an idea about whereabouts in Antarctica the vegetation is. But again, we've got this problem in Antarctic vegetation of density versus intensity. With the coarse resolution of the satellite, the Landsat that we used, you've got a 25-metre pixel in the colour band. And you're not sure in that 25 metres whether you've got total coverage of a lichen, which doesn't give a very high, which hasn't got much chlorophyll, much return in the or if you've got higher plants like Descampsia, but very patchy. They probably both give about the same results. So we needed a different approach really, because Landsat is never going to give you that, it's too coarse. Probably best to get better resolution both spectrally and spatially if we wanted to tell more about the vegetation. And also more ground truthing. And this is what we're looking at the moment. So one thing that we're doing at the moment, we're using infrared aerial photography to look at the actual discrete patches of vegetation to monitor change. There's not much infrared aerial photography over Antarctica. We have got very limited amounts, but luckily we've gone over the Leone Islands where we did our ground truthing. And you can see here one image that's been classified. So the red shows that on the infrared aerial photography, the vegetation shows as red. It's been classified on the bottom image, so that with classification technique to bring out the vegetation, which there is shown in green. It's about 50 centimetre resolution. This at least shows the discrete patches of vegetation, which the actual Landsats don't. So we've used this on two sets of aerial photography from 1996 and 2008 to monitor change on the Leone Islands. You have to look at the parameters. There are problems with the, it's not quite the same as using satellite imagery, using aerial photography. This is film-based aerial photography. It used to be scanned using the same parameters, and problems with sun angle and times of day. So there are some problems with it. But we can say that on the Leone Islands, the vegetation increased by about 36% over that 11-year period. Quite a large change. The next thing we're looking at is using hyperspectral data on Antarctic vegetation. We've got a plan this year to send down an ITRIS sensor suite, which has Cassini instrumentation aboard and some thermal capacity there, flying it over the vegetated areas at quite a low altitude. Using our Bass Twin Otters to do this, and we've got a plan to fly over the Antarctic Specially Protected Areas around Rothera, 6 of them, to try and map the vegetation. We're not entirely sure how well or what we'll be able to see, so this is quite, this is really, something we'll have to look into and see exactly what it will show us. But we hope to at least map vegetation type and extent. Of course, it's not just vegetation you can see with the hyperspectral suite. You may be able to say something about geology as well, and also do a little bit of species mapping, maybe on the penguins, as we were talking about earlier. So lots of opportunities there with the hyperspectral data. Okay, so 2 last examples I'm going to talk a little bit about. These are quite preliminary studies that we're going into, but again, quite novel. Ice seals. This is an example of looking for seals using satellite imagery in collaboration with the American NSF-funded Antarctic Geospatial Information Centre, using, I think in this case, QuickBird images. This is a panchromatic QuickBird image, 64 centimetre resolution. We're going to be searching for crab-eater seals in the Bellingshausen Sea along known transects. Now, the crab-eater seal is thought to be the world's most populous large mammal because it breeds on the sea ice, and there's huge amounts of sea ice in Antarctica in winter. But really that statement is something that's always been questionable, because nobody's really ever done a population count of them. They've looked at some transect data and extrapolated up from a limited number of transects. So we hope to be able to say whether that's true or not. Here we've got some lost seals from the Ross Sea. You can see in the little red squares, little red circles there, black dots which equate to seals. Now, if you cast your mind back to what we were saying about penguins and the criteria you need to look at when you're looking using remote sensing, we said, well, they need to be visible. Seals are visible, but they also need to be unique. Now, the seals aren't unique. This could be one of several species of seal. It could be a lost seal, it could be a Weddell seal, it could be a crab-eater seal. So they're not quite as good as emperor penguins for working on. But at least we can probably make some assumptions about the actual about the spread of the species if we get the data. So we're hoping to do that this year, and we will see what we get, and hopefully I might be able to talk on that at a later date. Another study that we're starting this year is measuring glacial flour in seawater. Glacial flour is fine-grained suspended sediments, usually made by subglacial meltwater along the base of glaciers. As it erodes the base of the glacier, it pummels the sediments into very fine sediment. And it comes, when it deposits it into water, it gives you this lovely turquoise colour, as you can see. This is Lake Louise in Alberta. And if you go to many lakes around glacial areas, you'll see these really bluey turquoise lakes. But also you can see it on the satellite images as well. Here's one of the South Shetland Islands. It's been stretched so that you can see these in the right bands. But you can see here, South Shetland Islands just on the northern side of the peninsula. On the northern side you've got this lovely turquoise colour. This is the sediment load in the water. Now,